So I think we'll start the session for today. I'll just share my screen. So please let me know if voice is first of all clearly audible or not, and if the presentation is clearly visible or not. Can somebody just confirm?
Sir, I think there is some kind of background noise coming.
Background noise. Yeah, I guess I'll try to just speak a bit louder. Yeah.
Yeah, no, it's fine, sir.
Yeah. Okay. Thank you. Okay, so again, I welcome all of you to the technically the official kind of final lecture series for the course Bioinformatics Algorithms and Applications by Professor Michael Cromiha. So this will cover the contents of week 12 class a little bit. And then what I plan for the majority of the session is that we will go over the earlier kind of concepts that we have discussed in this TA session, as well as that Professor Pomiha has also discussed. So in a recap style kind of discussion in a way, right? So we'll first of all try to see in the previous session what we did. We'll try a brief recap of that, and we'll solve a couple of questions that are maybe related to week 11 and week 12. And then we'll go with the recap for the previous kind of sessions for that. Okay. So yeah, okay. So let's begin with. So first of all, let me know what Professor Gomiha has went over or has gone over in the week 12's lab tutorial session or week 12's lecture series videos. I think this is supposed to wrap up the entirety of the bioinformatics algorithms and applications concepts, right? So what was the course content for that? Just somebody brief me on that.
Sir, actually, it was majorly an overview of all the week 11 till lectures, and also at last, it was a demo for VECA. I have not seen the VECA demo right now, but yeah, I have revised the whole weeks. There was applications and just a revision, I
Yeah,
think,
yeah, revision.
mostly.
So we'll go for that as well. And for Weka, I guess just watch Professor Gomiha's lecture, and if you want to kind of perform machine learning based calculations, so it's a good tool for that. But yeah, so okay, we'll start with briefly recapping what we went over in the last week's tutorial session, which was week 11, right? So firstly, of course, as we all talk about algorithms and models, so the evaluation of models in that part, so there was something called the ROC curve, right? So ROC was a receiver operator characteristic, and what the curve kind of plots is the uh, it's a graph between the false positive rate and the true positive rate, right? And we remember that if your ROC curve, for example, it's the area under the curve for the ROC curve, if it is 0.5, so that means that the model cannot distinguish between basically it cannot predict clearly. Okay, so that kind of indicates that the model prediction is occurring randomly in a way. So in that case, we saw that the true positive rate will be equals to the false positive rate essentially. So the true positive rate was basically a true positive divided by true positive plus false negative, and then the false positive rate was false positive divided by false positive plus true negative. So if both of these are kind of equal, so that means out of all of the true data in the set, the model cannot predict the model that is classifying it, the data points as correctly, truly predicted. So that value will be the same for the prediction where out of all of the false kind of a negative data in the data set, the model is depicting the data points as false basically. So in that case, your TPR will be equals to FPR and your model will be random. So that was kind of the FPR and TPR based concepts, and we also discussed some other terms related to the model evaluation. For example, your true positive rate is also called sensitivity, and the false positive rate we saw, but specificity was another particular parameter, which is sometimes called as true negative rate. So true negative rate is basically out of all of the negatives in the data, how many the model is correctly identifying as being negative. So it is true negative divided by true negative plus false positive on the denominator. On the denominator, you have the entire negative kind of data points in the data set, right? And we saw that your FPR or false positive rate and specificity has a relationships, which is FPR equals to 1 minus specificity. And then the other parameter related to model evaluation, which was accuracy, which is the correctly predicted divided by all of the present data set, data points in the data sets, which are all of the positives and negatives combined together. So that will give you the accuracy. So all of this combined, you have to use kind of in a way to assess your model. But mostly the area under the curve for the ROC curve, basically the receiver operator characteristic, that is oftentimes the best to evaluate your model. Um, in a lot of cases, for example, we just gave one brief example of classification problems where if you just go by accuracy, sometimes it's not good to just go by accuracy, whereas the area under the curve for ROC curve, that can give you basically a better picture of how the model is performing. Okay, and then in brief, we talked about something called the awk programming. So the awk programming was basically it's a command line kind of tool uh, by which you can parse through or you can collect some information from a large volume of data or data set in a very convenient way, basically. So we also shown an example of this particular of programming in Linux system using the terminal. So basically what happens that for a lot of analysis that you will do, you will have a large basically volume data set, right? So for example, we just gave an idea about how an MD simulation trajectory analysis data set will look like. So if you have for any molecular dynamics, if you have a large number of snapshots, for example, it's a 2000 snapshots. For each of those snapshots, you can have multiple column, basically multiple parameters in multiple columns, such as root mean square deviation, radius of gyration, several kind of distances, solvent accessibility, right? Different charge-charge interactions, even binding energy. For some cases, if we have calculated frame-wise, so all of those can be included in a large data set. It will be kind of a pain to go through it manually. So in that case, the awk programming can give us handy information inside what is written basically in the file by executing some simple commands basically. So we saw that you can identify a field or a column using a dollar symbol in the awk programming, and we looked at some of the kind of examples of that. For example, here we have the awk end print nr data.dat, which will give you the total count of lines in a data file. Okay, so count of lines is sometimes important. For example, if you're trying to merge 3 different or 4 5 different data sets and you are trying to combine them using a single command, let's say using some bash script in a single file, and then after that, if your data files have been combined correctly or not, you can find out by uh, taking in the total line number in the data file, the newly generated data file, because if you know the previous kind of files data points, so you just add them together, and then if the merging has occurred correctly, then your data files line number will be the addition of all of the previous ones, right? So in that case, it will tell you if your merging has occurred correctly. Another example was that if sometimes your analysis have been killed or have ended prematurely uh, during, for example, crash of your system or some power outage or something like that. So where exactly your data file has stopped, or where exactly your analysis have stopped, if your analysis output was kind of automatically being stored into a data file, so you can count the line number of that, and it will give you an idea about exactly at which point the calculation you need to restart again, right? So those kinds of things you can get easily using awk command line and this kind of basically commands. So we saw the other commands were basically kind of printing the data file into your terminal or into a specific um, basically text file that you can also kind of include in this command. For example, awk print fnr tab t $0. This will basically indicate all of the, or this will basically print all of the columns in the data file onto your terminal. So again, kind of handy if you want to include only some of the columns in your printed format. So that is also possible using $1, $2, or any of the columns that you want in your data file to be visible, basically. And then we went over some of the basically some of the numerical examples regarding the calculation of sensitivity, specificity, so on and so forth. Again, just basically you have to compare what was the actual value, which is oftentimes denoted as the experimental value, and what was the predicted value. And then based on whether your prediction is true positive, true negative, false positive, or false negative, you can just plug those values into this formula, and then you can get the exact value for any kind of parameter: sensitivity, specificity, accuracy, so on. Right. Briefly, we talked about correlation, and then we talked about neural networks. So basically, the architecture of a neural network will be one input layer, one output layer, and one or multiple hidden layers, whereby the nodes, you can say, of these different layers, they're connected by different edges, and these have some sort of weights which are important to kind of modify or manipulate in your parameter tuning. And also, I guess one of you said that it is possible without one hidden layer or without any hidden layer, there could be some sort of simple form of neural network, which is also called perceptron sometimes, but those will be only useful for basically simple problems, not more complex or more abstract problems. As nowadays we have seen that neural networks are being developed to process or solve, basically, right? So in this case, as indicated in the previous assignments, the answer should have been 3. And then we talked about a other form of representation of your true positive, true negative, false positive, and false negative, which is in terms of this confusion matrix. So basically, your actual values and the data points are being written down in the form of a matrix where you have the positives and negatives written here. And based on whether the data was, for example, actually positive and it was predicted to be positive, then you get the true positive value. If the data was actually negative, it was predicted as negative, then you get the true negative value, so on and so forth, right? So in that case, you have to indicate kind of which box in this particular matrix is which, and then from that you can get the TN, and then FP, FN, all of these values to calculate your parameters. So you can encounter these questions also. Just keep in mind that this is another representation that is quite possible in this context, right? And I guess we also went over this problem, very similar problem of identifying the sensitivity and specificity of algorithms. We looked over PDBTM. Yeah. So one of the questions that had also been present in the earlier assignments was PDBTM. So anybody knows what PDBTM basically gives your data on? So we know about protein data bank or PDB, right? So what is PDBTM in this case? PDBTM should be some additional form of kind of data included alongside PDB, right? So any idea? So it's for, yeah. Um, so option C, I think you are saying. So actually, it's particularly the TM stands for transmembrane. Okay. So it's basically transmembrane helical and strand protein. So in this case, the accurate answer would have been option D. Now, if you go for PDBTM, uh, as I was just trying to do, it will show, if you go for the original site link, um, it will show that the database has now been integrated into one other unified kind of database, which is called UniTM. Okay. So here, if you go through the paper for the PDBTM database, so you'll have the link for the PDBTM here, and as you see here, it's now been integrated into the UniTMP database. So if you want to go over transmembrane protein data, so this is kind of the database that we will go for. Okay. So UniTMP is Unified Resource for Transmembrane Proteins. You have the link for the exact database available in the paper also. And I guess it will open up the interface where you can perform searches and so on for different transmembrane helical strand proteins, etc. Okay. So I think it's kind of showing some lag in this particular link for the website. But anyway, you can go through the paper. for UniTMP to identify and look for different information regarding transmembrane proteins also. Okay, so just quickly wanted to mention that. Um, and yeah, so here in this particular question, the next one, so here we have again uh, some sort of talk related to algorithm, right? Now, if you have uh, an algorithm and it is kind of trying to assess protein stability, then the question is asking basically if you want to kind of develop an algorithm, so what type of algorithm would be ideal for this kind of analysis where you predict protein stability, ADL value, let's say. I mean, it's an addition here, but you have four options. One would be principal component analysis, one would be means clustering, one would be sequence to feature, and another would be linear regression. So for prediction of protein stability particularly, so which one should be, uh, I guess, the more appropriate option? So we have talked about all of these algorithms in some part of our earlier classes. So principal component analysis for those uh, who have used it, so you know that it's basically reducing the dimensions of the data set. Okay. So if you have a large dimensional data set, for example, again, I always come back to the trajectories of MD. So if you have large 16 dimensional data set, so what you can do is you can use the coordinate covariance matrix and you can reduce the dimension of the data set. So what this process basically does, the option A, it captures the variability in the data, and ultimately in the output you will get in a way where your first kind of eigenvector, you can say, the first principal mode, you can say, so that will capture the maximum variance in the data, and you will get an order of decreasing significance for these principal modes or principal components. So mode 1, mode 2, mode 3, and it goes on, right? And basically what it will say is that by analyzing the principal modes for the very first kind of the very earliest ones, for example, mode 1, mode 2, or principal component 1, principal component 2, principal component 3, you will be able to capture majority of variance in the data. So that will give you a good idea about what's going on in your data set. So that's a good way to analyze. It's always been analysis, a part of analysis, kind of in different molecular trajectory analysis, binding energy, molecular docking and simulation based studies also. And we saw that sometimes we basically utilize this sort of principal component analysis to create a Boltzmann distribution based free energy surface or free energy landscape of the protein. So that's quite useful. The next was k-means clustering. So k-means clustering we have talked about in one of our previous classes. So it's basically a form of dividing your data into a set number of clusters, right? And how it does is that firstly we will indicate so how many number of clusters you want, and then based basically from the available data points in your data set, so it will randomly assign one particular data point to be a part of the representative of a certain cluster, right? And then it will assign all of the other available data points in this particular data set onto a particular cluster. So on a basically into one of the already previously defined cluster ones where you already have an assigned data point based on the distance, you can say distance of this particular data points from the representative or the centroid of the clusters. Okay, so now that is one part of assignment done, and then it will calculate the overall variation in the data or the variance in the data. And then what it will do is that now the new cluster that is formed by assigning all of the data points, so it will compute a sort of mean position of the data, you can say, of the cluster. And then it will have that mean position of the cluster as a new centroid for the data. And then it will go for the assignment process again. So based on the distance, all of the data points will be assigned into again one of the particular clusters, or it will be part of one of the kind of assigned centroids. And then following the assignment, again, one iteration of your variance computing will be performed. And if the variance is kind of reduced from the earlier step, so that will mean that there is still room for improvement. And the calculation will go on again, one additional iteration will be performed. What happens is that if the variance is not reduced from the previous calculation, so that will mean that for that iteration, the k-means calculation is converged. Okay. Now from that you will calculate basically total variation and something also called within cluster sum of squares value. So those we will try to calculate, and then what the algorithm will do is that uh, in another separate set of calculations, it will start from different random points by assigning different random points into the clusters, and it will perform the same process again and again. And at the end of the process, the output or the result, it will compare with the previous sort of iteration, and it will compare whether there is an improvement or not. So in that way, if there was improvement, then the calculation will go on. If there is not improvement, then your calculation is stopped. Okay. So this will kind of go on for a set number of times, or this will go for sometimes until the entire calculation is converged. Okay. So that was the k-means clustering kind of. We saw that how you can assign your k-means clustering algorithm to particularly include this many number of clusters. So you can do that with something called within cluster sum of squares, and you can plot that against the number of clusters. And basically what you can find out is that in the sort of elbow-like structure for the plot, you can find out at particular point, at which particular point your WCSS reduction is being leveled up. So further increasing in the number of clusters, that will not reduce the WCSS value by very much, basically. So that will be your elbow point, or that will be the point, that will be the exact number of clusters that you need to include in your k-means clustering algorithm. So you saw an example of that as well. Now here we have seek to feature also. So seek to feature is basically, as the program says, or as the kind of terminology says, it's basically converting your sequence to feature. So you have given some sort of sequence, and then it will extract features from that. Okay. Now that's still not basically the prediction part included in this one. You can definitely extract features from sequence and use that to further build some sort of model that will predict protein stability. But that's not the whole picture in the seek to feature as well. So the answer that we are kind of looking at, the proper model would be in this case linear regression. Okay. So what linear regression is, you have one dependent variable and you have multiple independent variables. And what you want to do is that you want to predict the dependent variable from the basically available independent variable. So you build a model that can do exactly that. Okay. So in this particular case, for example, your protein stability will be dependent variable, and then the different independent variables, uh, those can be a n number of features, n number of kind of parameters. For example, your molecular weight, your long range order could be part of that, contact order, that details could be there. The number of alpha helices, the number of beta sheets that are present in this particular protein, that will be there. So different kind of structural features is what I mean to say. Different structural features could be part of the independent variables, and based on that, uh, you can try to build a model that will predict the protein stability, which will be the dependent variable. So that is kind of the linear regression part that will be coming. Okay. So the proper option in this case would have been option D, linear regression, right? And yeah, so we have talked about sensitivity, specificity, etc. We have discussed the formulas. So in this particular question, the next one, the question is asking that we have been given for a given for a particular method, the sensitivity and specificity values written 95.5 and 85.5 percentages, right? Now you are asked to uh, basically switch the labels in this model. So the positive class, that will now be the new negative class. So you are labeling, you are changing the labels from the positive class to be a negative class, and you are again changing the label for the negative to be positive, right? So what would be the value of the new sensitivity and specificity? So there should be some sort of change in the sensitivity and specificity values if you switch the labels, right? So what would that value, the new value, become? Try to answer it. Let me just look into it. Yeah, so we see option B here. So basically sensitivity was true positive rate, which was true positive divided by true positive plus false negative. Specificity was true negative rate, which was true negative divided by true negative plus false positive. And now what the question is demanding, that you switch the labels of positives and negatives, right? So basically what will happen is that in this case, your true positive will become a true negative. Okay. So true positive will become a true negative, and then the false negative will become a false positive. Similarly, on the other hand side, your true negative will become a true positive and the false positive will become a false negative. So now if you see, if you compare formulas of sensitivity and specificity, you will see that exactly if you switch the values from positive to negative, one becomes the other basically. So the sensitivity value will be changed to the current specificity value, and the specificity value that will be changed to the current sensitivity value. So in this case, the values also will be switching. So previously it was 95.5 and 85.5 percentage, and now the new sensitivity would be 85.5%, and the specificity will be 95.5% basically, right? So this kind of uh, small kind of logical type of questions you can also encounter. Just memorize these formulas, particularly for evaluating models, um, because Professor Gomi has also went over in detail all of these concepts, so you can encounter these problems. Okay, yeah. Again, a sort of kind of analytic type of question is here. So you have a new protein that is called protein X, and you are trying to predict, okay, which class this protein will belong to. So you have already two previously well-defined groups or classes of proteins, which are protein A and protein B, or group A or group B, and group B. And you have to basically decide based on the amino acid composition which of the following statement uh, will be true for analysis of this protein X. Okay, so you have been given four different statements. So one is asking that correlation between X and A is higher than B, and the protein should belong to group A. The deviation between X and A is higher than B, and the protein should belong to group A. The deviation between X and A is higher than B, and the correlation between X and B is lower than A, and as such, it should belong to B or all of the above. So just try to get to what exactly would correlation and deviation indicate. Uh, if your protein, two different proteins are similar, should their deviation be higher or correlation be higher? Or if the proteins are kind of different, then should their deviation be higher or correlation be higher or lower, basically, vice versa. So just try to figure that out and it should lead to the answer. In this case, of course, your correlation will sort of assume it is indicating a positive correlation. So positive correlation will mean that the change, basically, it's kind of it will indicate similarity. Let's just put it that way. Okay. So I see option C in the chat. So option C says the deviation between X and A is higher than B. So X and A, there is a high deviation. So of course, according to this option, the protein should belong to group B. But there is another addition in this statement also, which is saying that the deviation between, the correlation between X and B is lower than. Okay. So if the correlation between X and B is lower than A, so that means that X and A have higher correlation. So in this case, the statement is kind of contradicting. So it's saying that X and A will have higher correlation, but the kind of the first part will also is also saying that X and A has a higher deviation. Now correlation will indicate similarity and deviation will indicate dissimilarity, right? So in this case, the two parts are kind of not in agreement with one another. In case of option B, it says that deviation between X and A is higher than B and it belongs to A. So if deviation between X and A is higher, so that means X is very dissimilar to A as compared to B, right? So in this particular case, the protein should not belong to A, it should belong to B. Uh, in option A, it is saying that correlation between X and A is higher than B. So if correlation between X and A is higher, so that means X and A are the most similar type of proteins if you compare them with B, basically, right? That particularly in that sense, option A will be the correct one because it's correctly stating that if correlation between X and A is higher than B, the protein should belong to group A. Okay, so in the case option C did not have agreement with the two statements that was given, but option A, there is only one statement and it's the correct one. Okay, so in this case, the option would be A. So these kind of questions also keep in mind and be prepared for analytical type of questions also. So we have gone over sometimes functions, and basically they are implemented in all of the cases of where you need sort of mathematical part, right? In mathematical modeling, etc. In statistical analysis, also the usefulness of functions are there. So the question is asking that you have been given a function which is called fx equals to SGN of w0 plus w1 multiplied by x1 plus w2 multiplied by x2. Now we have been given the different values for w0, w1, w2, x1 and x2, and then the function has a condition for setting its value. Okay, so the SGN function of x, it will be 1 if x is greater than equals to 0, and it will be 0 if x is less than 0. So based on that, with the given w0, w1, w2, x1, x2 values, you are asked to calculate what will be the value for fx, basically. So just calculate this part, what is inside of the SGN function, and then apply the condition that is given here, that it will be 1 if x is greater than equals to 0, and it will be 0 if x is less than 0. That should give you the answer clearly. Okay, so try to calculate it out, and we'll just see in a moment. So yeah, option B you say. So as it is, you have to calculate the value of this w0 plus w1 times x1 plus w2 times x2. So it's of course 2 multiplied by minus 2, 2 plus minus 2 times 0.5 plus this will be 1, right? So essentially the value will be 2. Now the condition that is being given here is that if it is greater than equals to 0, so the value of SGN x that will become 1. Okay, so as it is given here. So in that case, since the value is 2, of course, the value of SGN x will be becoming 1. So that means the value for fx is 1. Okay. So just applying the condition that is given here, we can get the value of fx to be 1. Yeah. So another question regarding the uh, algorithms, it's asking that identification of co-expressing genes from mRNA expression data is an example of what? So you have some sort of mRNA expression data, and you are being asked to identify co-expressing genes basically, right? Now the options that are given here are binary classification, non-linear regression, clustering, and none of the above. So Of course, binary classification will mean that you are assigning some sort of data into basically a yes or no, zero or one or zero basically part, right? So that is kind of what a binary classification will be called. Again, regression, as we know, it's trying to fit uh, some sort of independent variables and trying to calculate a dependent variable from that. But in this case, it is saying that it's a non-linear regression. Third part is clustering. Okay. Uh, Now, in this particular case, basically what you can do, you can take the use of clustering. Okay. In the earlier case, we saw that we can build a regression model. But in this particular case, since it's talking about co-expressing genes, and if you have some sort of mRNA expression data, what you can do is that you can cluster them, and based on the similarity of the clustering being performed of the defined different groups that are being generated in the clustering part, so you can get the idea about co-expressing genes. So that should be the answer here, and as indicated in the assignment answers also in the earlier case. Okay. uh, and I guess one more question that we uh, will just go over. It is the R square. So we already have looked into the R values, right? Now R is the correlation value, right? The Pearson's coefficient of correlation that will tell you about the change in one kind of data set with respect to the other kind of data set, right? Uh, so if it is in the positive side, so that means they are positively correlated. If it's becoming on the negative side, then it's negatively correlated. So that's Pearson's correlation coefficient. But here you have been given the R square value. So R square value particularly, what is that called? Can you answer in chat?
Coefficient of variation.
So you said coefficient of variation. So coefficient of variation is not the correct one here. It's actually coefficient of determination. Okay. So it's the R square value that tries to capture the kind of proportion of variance in the dependent variable of a model, but it's not the coefficient of variation. It relates kind of the total variance captured with the data, and as well as with that how far each of the data point are from the particular level, I guess you can say, with that from the actual values, basically the experimental kind of set of data points. Okay, so it kind of relates both of that, and it is called the coefficient of determination, and it is a predictor of how good your model is. If it is close to 1, let's say, then your model is quite good. If it is 0.5, your model is kind of average, not that good. It's moderate kind of performing. If it is close to 0, then that means the model is not able to uh, perform its job clearly, basically, or the model is not that good. So you can try to improve the R square by tuning different parameters and playing with different kind of data sets, levels, etc. So that's one part. Okay, so I guess that will wrap up the entirety of kind of what Professor Kumiha has went over. And as you guys said, that a good portion of the week 12 was the revisit of the different concepts that he had went over in the earlier week sessions. So we'll again try to do that in a recap kind of discussion way. So yeah, so starting from the very first lecture, we talked about the briefs of bioinformatics. We talked about that bioinformatics is basically the broad idea of including all of the different scientific fields from biology to physics to chemistry, and the inclusion of maths slash statistics, because you have a lot of statistical kind of calculations, mathematics is needed here. Then there is computing and information technology, and combining all of that, you have the core kind of domain of bioinformatics, basically, right? So we said that in the different complexities of a physiological kind of environment, starting from your simple sequences to different structures to cells, cell signaling, tissues, physiology, organism, all of the cases there is applicability of bioinformatics. So can somebody give, for example, one quick example of how the protein sequences or the structures are coming into part of bioinformatics? So from protein sequence, what can we compute? Any example? So protein sequences, we saw that this can give us an idea about different parameters. For example, hydrophobicity, it would be one part, right? Different domains and their kind of informations, what type of amino acids are there, what are not there. So those kinds of informations we can get from protein sequence explicitly. So protein sequences can again be used for further calculations of protein structure. Now, protein structure is kind of a bit more complicated, where you have the 3D information of the biomacromolecule at play, basically. So, for example, in protein sequence, we could have calculated the hydrophobicity of a certain region of the protein, right? Now, in the protein structure, if it is coming into play, we can not only calculate the hydrophobicity of some kind of region, we can also calculate the surrounding hydrophobicity. So in this case, the surrounding will be what is coming in close contact or in close proximity with a particular region in the 3D space, right? So now you have the different atoms and their positions. Now you can calculate the contact that have been formed in the particular structure. Now you can calculate different distances. Now you can go for basically your long range order, contact order, these kinds of calculations. And ultimately, you can also go for studies of protein folding. If you have the protein kind of structural information, you can go for the conformational changes. So those kinds of, again, more kind of complicated calculations you can perform if you have the information about the structural data, which again, another part that is directly kind of feeding into, that is protein interaction. So protein interaction, it could be interaction with any of the kind of physiological elements that drives forward cellular signaling. So again, as you see, that if you are able to interact, if you are able to calculate protein interactions, the different biomolecular interactions inside of a cell, you are directly getting information about cellular signaling. And those cellular signaling, if you go for a bit more complicated kind of environment, it can give you information about tissues, organs, physiology, kind of any sort of setup which you can think of that is possible biologically. So it can give you information on that. We saw that the application of bioinformatics was ranging from structural analysis to sequence analysis to functional analysis. Where in the case of structural analysis, you have the nucleic acid structure prediction, protein structure prediction, structural classification, as we saw, structure comparison, of course, that is also a part of 3D kind of structural computation. Structural classification, we saw that based on the different types of, um, for example, uh, secondary structures being formed in the protein. So you can calculate the proteins based on the all alpha, all beta, or alpha by beta or alpha plus beta kind of category, right? So based on the dominance of the alpha helices and beta sheets and so on. So those kinds of, you can get the structural classification also. And as we saw that directly leads or that directly kind of correlates with some of the other values that we have also kind of calculated earlier. For example, contact order, long range order, those kinds of values, and as well as folding rate. So if you are able to classify the protein, you can have idea about its behavior in different settings also. On the sequence part, we have the genome comparison, phylogeny, promoter prediction, motif discovery, sequence alignment, of course. And on the functional side, you can model metabolic pathways, you can profile gene expression, you can have protein interaction, as well as subcellular localization prediction. So there are different tools for that. For all of these cases, you can look forward on the internet. Nowadays, different AI-based tools are being developed also for all of these categories, basically, including something, for example, functional analysis like subcellular localization prediction also. You can get a good idea of what is um, going on there. And just combining all of these, as I said, there is one part which reads as software development and database construction and curation. So software development is basically you are developing a tool that will help others performing the same analysis that you are doing, right? So that is the software development. And database construction and curation is another part where you are taking already available data that are being generated by different researchers across the world from different scientific groups, and then you are taking all of that together. You are putting them in a you are putting those in a database in an organized way, so that the data retrieval and data access becomes more convenient, basically. So that will save time for future researchers who want to who wants to basically access your data or not your data, all of the available data for some sort of study, right? That will make it very convenient for them. So uh, database construction is quite important in that case. And of course, the significance of bioinformatics ranges from functional genomic studies to computational studies for protein ligand interactions to again healthcare related issue fixing, kind of forensic science, agricultural benefits. So all of these cases, it is applicable very simply. And the five kind of core part of bioinformatics on the side of application based. So Professor Bromia that he mentioned. So one is database construction, two is derivation and testing of hypotheses, three is web servers. So web server is basically a tool that is available on the web where you can perform different sort of analysis. Fourth is virtual screening drug discovery, which we have talked about. And the fifth is the big data analysis, which is the next gen sequencing, right? We talked about different databases and how we can retrieve different important information from those. For example, one of the very important crucial database for proteins, which relates to protein structural information, that was RCSP-PT, protein data bank, right? Where you can get sequence related information, DNA related information, etc. Those sequence based informations are available in Gene Bank, DNA Data Bank of Japan, and EMPL. Right. So these are sequence based kind of databases. Then what we basically talked about is that so a big part of analysis is trying to understand whatever sequence you have or structure you have, and trying to calculate different parameters that will give you insights into those sequences or structures, right? So we talked about one very simple sort of parameter, that is the composition of uh, certain type of amino acids in a particular sequence. So you can get useful information from that as well. So for example, this question was asking the composition of positively charged amino acid in a particular sequence, right? Now, based on if you have a high positive charge content, so you can identify kind of that particular region to be interacting with some sort of negatively charged component. So this sort of information you can get. You can get DNA binding proteins in the same kind of logic as well, DNA binding regions of a protein in the same logic, using the same logic as well. And basically, very simple set of numeric calculation where you assign or you try to figure out which of the amino acids of the sequence is positively charged, in fact, and you just divide that by the total number of amino acids present in the sequence, multiply by 100, and you should get this kind of answers for the numeric questions. Yeah, so that was one quick question that we went over, and then we went over uh, briefly about next gen sequencing. So the primary point was that the next gen sequencing methods, so not only they are generating huge uh, volume of data sets, but the next gen sequencing methods, for example, Illumina NGS, they have made possible for the sequencing to be performed at a very low cost, and the performing the basically the sequencing time that has also reduced by a lot basically. So here one plot we discussed that shows the cost per raw megabase of DNA sequence. And as you saw that around the time of 2007, there is a drastic change in this plot, which if you compare with the Moore's law, it will show you basically a rapid advancement in the field. Okay. Now the Moore's law is basically kind of a continuous advancement in the field by comparing with the advancement in the computing power, which is kind of the transistor systems. So the number of transistors in an integrated circuit that doubles every 2 years basically. So if you plot that graph, you get this sort of Moore's law slope. And if you have a rapid advancement, you will see at some point that the plot for your raw megabase per DNA sequence cost, that is decreasing rapidly, and it is leaving the Moore's law far behind. So that is what where the advancement is kind of starting to show basically. Okay. The same thing is observed for the cost per human genome sequence, where you see at the beginning of 2001, so a lot of zeros are after this one in this kind of dollar value. But if you see where the plot is ending kind of around 2022, so at that point, you can basically sequence your genome at $1,000. Um, So the cost has reduced by a lot, and it is again following the same uh, trajectory as the previous plot basically. And in this particular plot, if you see at any particular point where there is a valley or a rapid decrease in the cost, so in that particular point, that means some sort of new advancement in the next gen sequencing methods they have been introduced. So that is why there is a sudden decrease in the cost at that point. Okay. So what does NGS have to do with bioinformatics? Of course, for analysis of huge data sets, massive data sets that are being generated, you can have the tools for genomic assembly where you can have reconstruct complete genomes from short DNA RNA fragments. You have functional annotations, comparative genomics, transcriptomics, epigenomics, and personalized medicine. All of these are coming under the NGS and bioinformatics umbrella, where you can get the sequence data and you can compare or you can perform different calculations which will be useful for all of these cases basically. Now you saw that the sequence, if you try to calculate some parameters, for example, from a trinucleotide or dinucleotide sequences, uh, so it is possible. But before that, you have to first try to identify uh, which are the available, which are the possible trinucleotide or dinucleotide sequences which can be uh, obtained from a given kind of sequence, right? A given small sequence, you can say. So if you calculate these parameters in small stages, in dinucleotides, and if you add them up, so that is one quick way of getting the overall parameter value for that particular small subsequence, let's say. Now, how you do that? You basically go for uh, overlapping dinucleotides and trinucleotides. Okay, so by overlapping dinucleotides or trinucleotides, that will mean that if you have the first dinucleotide as AG, then the next dinucleotide you do not count from C. Okay, you do not count from the next one, but you also include where you stopped in the previous step. So if you count, if you have the first dinucleotide as AG, the next one will be GC, the next one will be CA. So one overlap is already always staying between all of those. And the same case for trinucleotides. So if you have AGC as the first trinucleotide, the next trinucleotide you will have is CAT. The next one will be TGT, etc., etc. Right? Now, why you do kind of overlapping? Because if you do not do overlapping, then in a lot of cases, local information goes missing somehow in some kind of points. Okay, and also you will have a higher number of basically dinucleotides or trinucleotides if you go in overlapping fashion. So that will improve your statistical uh, significance of the calculation as well. Okay, so from this quick formula, if n equals to 13 for the 13 different nucleotides or different protein, different amino acids you have in this particular sequence, so you can calculate the number of trinucleotides as n minus 2, you can calculate the number of dinucleotides as n minus 1. And if you calculate the dinucleotides and trinucleotides, and if you have the available information regarding some sort of parameters, as in this case is hydrogen bonding energy. So for the dinucleotide AT, it's given as -7, for TA it's -7, for GC -12, so on and so forth. So you can calculate this particular parameter like hydrogen bonding energy for the entire stage of the sequence. Okay, that's what you can do if you have the dinucleotide or dinucleotide information available. So in this particular question was we had the subsequence divided into dinucleotides ATTA, AGG, and AT. And by plugging in all of the values that have been given and by doing an average, you get a value of -7.8 for the average hydrogen bonding energy for this small subsequence basically. So that's also possible. We saw very similar calculations for different parameters, again hydrogen bonding energy. And then we also went over some similar concepts, for example, GC content, right? So GC content, if you have, if you are encountering this kind of problems, you just do what you just do is that you identify all of the G's and C's that are given in the sequence. Again, you count them up and you divide by the total number of nucleotides that are present, multiply by 100, you should get the answer. So basically average energy, hydrogen bonding energy, calculation of composition, and then also calculation of GC content, for example. So these are the few calculations that you can encounter in the exam based on the earlier sets of uh, basically problems that Professor Kumi had discussed. Okay. In this case, we also saw the sequence-based parameters like GC content that can be calculated using uh, the small, very simple formula, right? Now, sequence-based parameters are basically different parameters that you can obtain from sequences, as we said. On the protein side, we saw that we can calculate contacts, we can calculate, not contacts, sorry, you can calculate compositions, we can calculate hydrophobicity, these kinds of parameters. On the DNA side, of course, you have DNA rigidity, thermal stability, base stacking energy, hydrogen bonding energy that you can calculate directly from the sequence, of course. If you have the information of those parameters in the terms of dinucleotides and trinucleotides, as we discussed earlier, right? So basically, uh, the number of nucleotide steps. So this particular slide is basically talking about uh, which uh, number of nucleotide steps you should include in your calculation for different parameters. For example, you have just information regarding mononucleotides, or should you have information regarding dinucleotide sequence that you want to include, or should you have information, or should you include information regarding trinucleotide sequence? Okay, so it's saying that the number of sequences or unique sequences or steps that increases as we go from mononucleotide to trinucleotides. Information also increases and available data decreases for some steps slash sequence. So that's one basically caveat of using this sort of small nucleotide stretches for calculations. So basically, what the point is trying to tell that, of course, if you are, for example, using overlapping dinucleotide or trinucleotide steps, you will capture a lot of local information. You have more number of sample data, so your information overall should increase. But what oftentimes have is that if you go forward, so from trinucleotide, if you go forward and let's say you are trying to calculate the parameter using tetranucleotide sequences and even pentanucleotide sequences, let's say, right? So what will happen is that, of course, the logic says that if you have a small stretch of sequence, right? Now you are trying to calculate some sort of average parameter for this using pentanucleotide sequences, right? So that means that for those pentanucleotide sequences or for all of the pentanucleotide sequences that are possible, you need to have the parameter values already calculated or computed beforehand. That is how you will add all of them up, and that is how you will do the average calculation, and you will get the value for your entire stage of sequence, right? Now, if you go for tetra or pentanucleotide, it can happen sometimes is that for certain pentanucleotides or certain tetranucleotides, you do not have any calculated values or computed values or even kind of experimental values, right, that are available. So in that cases, the availability of data for certain uh, tetra or pentanucleotides, that is gone basically. So that will hinder your calculation for uh, the overall parameters using these kind of sequence steps, right? So it's better that you follow either dinucleotides or trinucleotides. They will capture the a good amount of information, and they will also have they will also not encounter these sort of problems where it might be that some sort of in some sequence steps you do not have any information available for this. And in this particular case, kind of similar thing is shown in the chart form. So you see that here in DiPro-GB, this database, you have dinucleotide sequence and their related parameters calculated from for these different kind of properties. Okay. So for twist, stacking energy, bend, inclination, major group, minor group, width, etc., melting temperature. So all of these values you can calculate, or all of these values have been calculated for the dinucleotide steps, and they are included in this DiPro-GB database. And then on the other hand side, you also see that Young's modulus, or which is kind of indication of DNA stiffness. So that is being calculated or that is being computed for the trinucleotide sequence. Okay. Here again, you see that the exact same problem, the number of sequences or the number of trinucleotide steps for which the Young's modulus calculation was done. So you see for some of these cases, you only have a very limited number of data. Okay. So for example, here you only have one sample for ACA, TGT, this one, right? So of course, it means that if you go even forward for tetranucleotide, pentanucleotide, you will have a lot of steps for which you do not have any information. So you cannot use those for computing any parameter for a large set of sequence, right? So that's kind of the point that was mentioned in earlier. Again, same problem is discussed in this question. Number of dinucleotide and trinucleotide sequences that you can get. Just remember the formula. Trinucleotide means n minus 2. Dinucleotide will mean n minus 1. Okay. Similar question, average hydrogen bonding energy. Simple, just divide the sequence into the dinucleotide or trinucleotide steps as needed, and then you can just add the values, all of them up to get the total value, divide that by the total number of sequences generated, and then you should get the information. Now, the databases we talked about briefly, which is basically an electronic repository where it is facilitating quick retrieval of the data from a organized kind of set of collection of the data, right? So the organized set of collection of the data, which also facilitates easy retrieval on filtering based on different criteria. So that is basically, in simple terms, a database. So it has the both hardware and software component for data management, and the goals are basically to structure data into records, and each contains multiple fields. For example, names, phone numbers, addresses, different kinds of fields can be there. And when you search, you want to retrieve some information, so you can use something called the query to the database to retrieve the specific values or specific records there, right? Now, there are some terms which are kind of part of this whole database discussion. One is content, which is what is stored in the database, and there is ontology, which is the valid terms and their definitions, the logical structure, also known as schema. So schema is the term that you need to kind of memorize more in this case. So that is the interrelations amongst the data, that representation of that. And then you have the format of the data, you have the retrieval methods, and you have links, of course. So links are the links to external resources, which are kind of supporting certain parts of what is there in this particular data point. It could be publications, it could be tutorial materials, it could be links to other databases also. So that is what database kind of is broadly. And then for the biological databases, you had three types of database, the overall classification. So one was the primary database, which you have GenBank, GenBank, then EMBL database, then your DNA Data Bank of Japan, as well as Protein Data Bank (PDB). So those are kind of examples of primary databases, which basically store original biological data. They have raw sequence or structural data that are submitted by the researchers. Okay, so that's the primary databases. Now the secondary databases are sometimes processed or curated form of the primary databases. So they are derived from the primary databases, but they go through a manual curation step. And they often include different functional annotations, and the examples are SwissProt and protein information resource. Now we can also have specialized database, for example, ribosomal database project. So this is again related to a very specific or niche area of research, for example, ribosome here. Okay. So these sort of different databases are possible, and for development of databases and for accessing and performing different tasks on the databases, you have a programming language that is called SQL or SQL, which is called structured query language. So we looked very briefly into some of the commands of SQL, and I believe that I gave basically one link to a small tutorial or small website where you can basically access the different simple commands, and you can execute them to see what they do in the case of different programming languages, right? So what structured query language is? It's a specialized programming language for manipulating relational databases. Now, the relational databases we will see very briefly. So what SQL does is it's a standardized way to react to the database regarding basically any kind of database management system that it uses. You can use SQL to interact with those. Then it can be categorized into DQL, data manipulation language, or DML, and data control language, and some simple SQL commands I just mentioned here. And this is the particular online tutorial that I showed, W3 Schools. So you can have different commands execute the different commands on their website terminal, and then you can see what tasks is being performed. Okay, so the relational database term was being mentioned just earlier. So the relational database is basically data present with different tables with at least one common attribute. If we have this kind of database structure, so that will be a relational database. Now, the database management system overall, it is the program that is uh, being utilized for the streamlining the organization search retrieval of the data. So that is the database management system, and the relational database management system, that is one part of DBMS. The other one, as we talked about, that is object-oriented database management system. Right. Now we talked about uh, the particular structure of relational databases or the different facets of relational databases. So firstly, the structure. Now there should be one common column or field between the databases. So of course that means that the data is organized in a form of a table, right? In a not only a single table but multiple tables. And if you have multiple tables in that sense, each of the table they will be termed a relation. Okay, and the tables of course are inclusive of different rows and columns, where you have the different fields indicated in different columns, and the rows will be the specific values according to the fields, which will give you a particular record for the database. Now the indexing, they are based on a shared characteristic which is called an attribute, and there could be cross-referencing also. How you execute query is basically the system select linked data items from different tables during query execution. So the linked, the common basically attributes or the common fields that you have between different tables. So those will be accessed to efficiently go through the entirety of the database. Basically, that is what will facilitate a quick access to the information. And it has advantages over flat file database, because flat file database, where you do not have any hidden instructions or where the data is not organized in a particular fashion. So in that case, you have to go through the entirety of the file to find certain information. So that is oftentimes very computationally uh, costly. You cannot find a suitable kind of solution for finding out or basically getting certain information from a flat file database. So that is why SQL is being developed. SQL has been developed to basically organize in a relational database. So an example we had given earlier. So in this particular case, the flat file database has name, states, course number, course name, and there is a delimiter or a pipe that is separating the different kind of fields, not the different fields, but the different data points from one another. So for example, the first one is John Smith, Texas, Biology 689, Bioinformatics, etc. Right. So what course they have taken, what is the course name, so on and so forth. Now, if you are asked that, for example, from the state of Texas, how many people are getting the course Bioinformatics, right? Now, if you have a large flat file database, this is very small what is given here. But if you have, let's say, thousands upon thousands of pages in a database, and if you want to go over the entirety of the database to find this information, of course, that is not possible, right? It is computationally very, very expensive to do that. So in that case, what you can do is that you can divide or you can organize your database in different tables. Now, if you look into the tables here, you will see that certain columns are common between different tables. For example, in table A, you have the student serial number as well as the name and state. In table B, you also have the student serial number, but it does not have the name and state. It only has the course, right? In table C, you have the course, which is common with table B, if you notice, and then you have the course name. Now, if you are being asked to perform this calculation, for example, how many from Texas are taking the course Bioinformatics, so you just have to go through the entirety of table A first, like, right? So only the name and state information and the serial number, of course, you have to go through that, and from that you will basically get an idea about what to do in the next step. For example, from corresponding to the Texas state, you can get the student ID serial number 1 and 5. For the next kind of table search, you do not have to kind of do not have to perform an exhaustive one. So you can just see that corresponding to student 1 and 5, um, basically which courses are there basically, right? So corresponding to student 1 and 5, you will see one is only taking the biology 689, which is bioinformatics, as can be seen in table C. So in this case, for table 2 and table C, you did not have to do an exhaustive search as you had to do in the case of a flat file database. So that is kind of the advantage here. So it reduces the computational time taken in this type of organization, and the relational databases, they have the benefit of SQL or SQL, right? So they have hidden instructions which makes it possible for executing different commands and retrieving different data from that. Now, another type of database that has been discussed was object-oriented databases, which are developed using different object-oriented programming languages, for example, C++. And there are also some sort of hybrid approach in the case of relational and object-oriented databases and combining both of them together. So the object-oriented database kind of looks like this. You have a pointer, you have different objects and different kind of blocks which sort of represents a particular data point, let's say, and these pointers kind of connect all of these different objects together. Okay, and from that one, by tracing which pointer is being connected from which object to basically a certain object of a certain field to a certain object of another field, by tracing that, you can ultimately get any information that you want from an object-oriented database. So the coding and how it is basically works, you can look it up. So you need to firstly go through the concepts of object-oriented programming language. In this case, some examples are given here. For example, C++, that is a object-oriented programming language that can be used for different tasks. And then you can get the idea about the codes, how that basically works, right? Okay, yeah, so we also went over computational biology, which was one of the kind of 5 pillars that Professor Gomia mentioned for bioinformatics. And we saw that computational biology was useful for prediction of anything biology related, right? So it could be predicting potential lead molecules, it could, or identifying potential lead molecules. It could be prediction of different biological affinity values or data. For example, IC50, it could be prediction of different ADMET parameters, toxicity, absorption, distribution, anything you can predict. We looked at different web servers that already do that. For example, Swiss ADME, ADMET Lab, and so on. So the usefulness in this case is related to prediction, what we can predict from the available data. Okay. Yeah. And then we went over the sequence alignment process, right? So sequence alignment, of course, what it is basically aligning 2 or more protein or nucleotide sequences. And this can give us very important insights into phylogeny, as well as can help us into different other areas. For example, motif discovery. It can again give us idea about the structural part also. So for example, we know that for homology modeling, etc., you also need to perform sequence alignment. So the importance is wide range basically. So there are essentially 2 types of alignments. One is called the global alignment, and another is local alignment. So global alignment, you try to compare the entirety of the sequence. For local alignment, you try to focus on particular parts, small regions or part of the sequence, which could be corresponding to different conserved regions or motifs. So that you only focus on, you try to maximize that score only. Uh, so that is for local alignment. It's useful for functional annotation, structure prediction, etc. Because small motifs and functional motifs are basically what you are getting at with this kind of alignment, right? And now for sequence alignment, um, the very simple visualization tool or plot that we went over, that was called a dot matrix. Okay. Now, dot matrix, of course, as it is saying, it's introduced in 1970. So the approach is basically construction of a graph where you have on, let's say, the x and y axis, or it could be, for example, across the page, another down to the left hand side. Uh, you have these 2 sequences written, and whenever you find a matching character between both of these coordinates, basically x and y, So you put a dot at that intersection point in the graph, and then when you do kind of end up placing all of the dots together, what you try to find, you try to find particular diagonals. Okay. Now the diagonals in this case, they will reveal the regions which are aligning together basically. So from that you can get ideas about different stuff. Now how it works, so this was an example of dot matrix. For example, on the right hand side you have the dot matrix for this lambda ci and P22C2. So if at any point you have a diagonal, for example this particular region, they seem to be showing a diagonal here, right? So that means that for this range, if you project that onto both of these axes, right? You project that onto the x-axis and the y-axis. So the range that you will get, so that will be the part that is aligning basically. So that's how these diagonals basically work. And we saw with a brief example of how to do that. So for example, you have ACG-CG versus AC-ACG. Now you try to match one by one. So if it is, for example, A versus A, so you will put a dot here. In this particular case, it's A versus C, so there will be no dot here. Only in the case of similarities when you put the dots, right? So A versus A, G versus G, C versus C. So that's what you are getting at. And when you finish constructing the plot, you will see that in this particular region, there is a diagonal, right? So this A versus C, A, CG, CC and GG, so they ended up creating a diagonal. And interestingly, if you project that onto both of these reaction coordinates, you will see ACG is the part that is common among both of these sequences, okay? So with a window size of 1 and a stringency of 1, you could get this value and you could overlap the ACGs together. So they are the matching part. But sometimes what can happen is that if you have a large sequence, a very large sequence, so if you try to match one by one and if you put all of the dots there, the dot plot can be quite messy. So in a lot of cases, you cannot figure out exactly where a certain diagonal is. So in that scenario, what you do is that you increase the window size and you place some sort of stringency value to the calculation. Now, what a window size is basically, it's saying that instead of putting a single character by single character match, now you try to compare three different characters of the sequence together as a single window. So that's your three character window. And if as per the stringency value of 2, if you have two exact character matches at the exact same position, then you put a dot between these two windows. For example, here we have a ACG versus ACA, and we see that in both of these cases, the initial two characters are A and C, right? AC here, AC here also. So that means that with a stringency value of 2, so that means at least two characters are matching in this three-step window. So you can put a dot between these two three-step windows, ACG versus ACA. That's what is being said here. Then you go for the next kind of, if you try to find out the next set of dots where you can put those. So CAC versus CGC, again, CC on the first and third, they are matching. So you can put a dot here. And ultimately, you can end up with a very reduced number of dots on the graph. And what this will basically do is that it will help you clear the mess of all of the dots, kind of, if there is a large number of dots. And sometimes if you try to manipulate this window size and stringency into different values, you can actually identify the windows or the diagonals and where basically they're being created, okay? And we also discussed the different tools. For example, this is one tool from Barry Grant at the Grant Lab. So they also develop different visualization analysis tools. For example, in RStudio, they have developed Bio3D, which is a good tool for different calculations such as network dynamic cross correlation and different kind of calculations. So they have made a visualization tool on the website that you can play around with, and it will basically help you understand the dot plot, right? Yeah. Now, as I said that reducing the number of dots, they can help you find out the pattern that you are seeing in the data set, right? So, for example, if the cluttered plot was looking like this, So a lot of dots are present here. Now if we apply a certain window size, so it is saying that window equals to 23 and stringency equals to 7. Now all of a sudden you have a very clear sort of plot. Of course, the main diagonal is here because this is a same versus same nucleotide sequence comparison. But what you can see is that there is a repeated pattern of small stretches of sequences that is being seen in this case, which is why in the same sequence for the same sequence dot plot, you are seeing this diagonals that are forming at this sort of way, right? That means that repeats are present in this sequence. So again, you saw that reducing the number of dots by increasing the window size and stringency, that helped in clearing up the plot and getting the inference basically. And for sequence alignment, of course, dot plot just provides a simple visualization process, right? What it does not do is that it does not explicitly quantify how good the alignment is. For that, you need to introduce a scoring system. Now, if you talk about simple kind of scoring methods, for example, in this question, so they have given a match score, a mismatch score, and a gap score. Now, a match score, as it says, if you have the two characters of a sequence matching, so you will get the match score. And it is a positive score because, of course, the match, if there is a match, so that means your sequence alignment for this particular character by character, that is good, right? That's why you are giving it a positive score. A mismatch here is given as -1. So that means that the characters are not matching, right? The sequence characters are not matching, so that is why it is penalized. And a gap is given as -2. Now, what happens that in nature, of course, uh, introduction of gap is much rarer. So that means that in your sequence alignment, if you have a gap, so you have to give it a higher penalty, even higher than a mismatch score, because it's possible that since it's the gaps are kind of not favored by evolution a lot of times. So if you have a gap in your calculated sequence alignment, so it could be that the gap was the calculation for this particular part was not what it should be. That's why it is penalized basically. A gap is penalized. So the gap normally should not be there, kind of, right? Now, depending on the matches, mismatch, and gap scores, of course, you can easily calculate what is the net alignment score for any sort of given alignment, and as in this case, it is -1. And sometimes it can happen is that instead of one gap penalty, you can be given two gap penalties, which are origination and length penalties. Now, the origination length penalty is self-explanatory because how many gaps you have, that will be the number of length penalties that you need to give. Now, the origination penalty is each new origin point for a gap. So, for example, on the left-hand side, one gap is originating here and another gap is originating here. So, it will have two origination points for gaps and two origination penalties. However, on the right-hand side sequence alignment that is given here, so only one origin point for gaps are there, and the next gap is just subsequent to the previous one. So that is not counted towards the origination penalty. Okay. So in this case, it will only have one origination penalty. Just remember that. And again, the scoring calculation is very simple numerical calculation. So based on the best scores that you get from your alignment score calculation for the different alignments provided, so you can get the best alignment possible. So that was basically the origination of gaps and penalties. And now we talked about the different matrices that Professor Kumiho went over, for example, PAM matrices and Blossom matrices, right? Now these are basically the matrices that will help you identify what is the kind of the relative mutability or kind of the amino acid staying the same, the propensity for that in a matrix format. Okay, so PAM matrix was constructed by Margaret Dayhoff with 71 groups of closely related protein sequences, and it's termed as point accepted mutation, right? So they took greater than equals to 85% similarity, so very highly homologous protein sequences, and if you have in this calculation, so if you have observed any amino acid mutations in the data, so that amino acid mutations, those are being called as accepted by natural selection, right? Now, first what they do is that you have to calculate basically a PAM1 substitution table. So based on the mutation frequencies from one amino acid or one sequence to another, one amino acid to another, you have a PAM unit of single PAM, right? So one PAM unit can be defined as 1% of amino acid positions that will have undergone changes. And then for the calculation of PAM2, PAM3, and any of the subsequent sort of PAM values, what you have to do is you have to go for matrix kind of extrapolation, matrix multiplication basically. So you have to extrapolate that value. You do not calculate that value specifically. So if you basically multiply PAM1 by PAM1, you get PAM2, right? If you multiply PAM1 with itself a number of times, you get the value of PAM for that particular kind of time. For example, PAM42 will be PAM1 times PAM1 times PAM1, 42 times in total. Okay. Of course, in all of these cases, you do a log odds score computation. You interpret the matrix code as the positive scores indicating that the substitutions occurring more frequently than anticipated, and the negative scores indicating the substitutions occur less frequently than anticipated. Right? You have a corresponding observed mutation rate as well as sequence identity for the different PAM numbers. For example, PAM1 will be, of course, 1% mutation rate and 99% identity. 30 will be 25% mutation rate, 75% sequence identity. 250 will be 80% mutation rate, 20% sequence identity, and so on, right? So that means if you want to perform sequence alignment, and you want to retrieve the scores for a certain character by character match or mismatch from the PAM matrix. So if you're trying to calculate alignment between closely related proteins, it's better that you go for a lower value of PAM. For example, PAM40, it could be, right? If you are trying to calculate the or if you are trying to do sequence alignment for highly distant proteins, then you should go for higher value of PAM. So the value for the amino acid by amino acid kind of mutation that you get from a higher order PAM matrix, you can include that in your sequence alignment, right? So something like PAM 250 looks basically like this. You will see that all of the same amino acids, they are kind of forming this diagonal. They have a positive value. Uh, of course, the positive values, if you compare like the different same versus same amino acids, so it means that not all of the amino acids have same propensity to be similar, right? To stay the same because they undergo mutation, of course, right? So the lower positive values, that means that these amino acids are going, these amino acids are very prone to be mutated, basically, in that sense. So in that case, tryptophan and cysteine, so these will be the amino acids that are more resistant to mutation in a common kind of way. And then for certain amino acid to amino acid transition also, you can get the score here. So if it is, for example, a positive score, that means that this transition could be favorable. If it's a negative score, so that means the transition is not favorable, right? The mutation is not favorable. So you can get that idea from here also. So that was PAM matrix, and PAM matrices, of course, they were developed from direct observations of closely related protein sequences. So that means that for the distantly related protein sequences and so on, so you did not have basically direct observation based values, right? So you had extrapolated values that you are deriving from PAM1 matrix by matrix multiplication. So to address that, of course, Blossom matrix was developed. So Blossom is blocks amino acid substitution matrix, and they are constructed based on every possible amino acid substitution in multiple sequence alignment. Okay, so as compared with PAM, uh, it's kind of the opposite in a way, uh, in the sense that if you are trying to calculate closely related protein sequences, you usually go for lower PAM values. But if you are getting that from the Blossom side of the matrix, so you should go for a higher Blossom value to include sequence alignment. Okay, so kind of reverse in that order. So the same is given here in the case of Blossom matrix. Blossom 62 is mentioned here. So again, you can see similar behavior. For example, cysteine versus cysteine is the most kind of conserved or resistant to mutation. Similar amino acid to similar amino acid transition, you will see they will be favorable. For example, a charged amino acid positively charged one like lysine, if you compare that with the, if you compare that mutability with the positively charged amino acid arginine, you will see that it has a value of +2, right? that means that is favorable. Again, based on the characteristic of the amino acids, you can also get to the same conclusion basically. And as I said just earlier, so for closely related sequences, the most suitable one would be some lower value of PAM like PAM 40 and a higher value of BLOSSOM like BLOSSOM 80. And the sequence alignment, in the sequence alignment part, we went over the Needleman-Wunsch algorithm, right? So the Needleman-Wunsch algorithm is basically uh, the calculation of a kind of it is basically where you have a matrix kind of setup. On the left hand side and on the upper hand side you have the two sequences mentioned, and then you have to fill up all of the boxes inside of this square by using this match, mismatch and gap scores. Now you do an initialization with the value 0 on the top left corner, and the rule is that if you go horizontally, it will indicate a gap. So if you go in this particular way, which you can do using the gap score of -2, so you will continuously build up the gap score one after another. For example, the first um, box just besides the 0 value, so it will have a value of -2, and the next one will have a value of -4 because the gap score is being accumulated. Okay, and on the first column, basically, again, the vertical jumps, they will also indicate gaps. So again, here also the gap penalty will be accumulated. Now what you have to do is that based on a particular match value or mismatch value, you have to calculate uh, the potential value that can be put in all of the other cells. Okay. Now, what value will be that? So it will come from this algorithm. So you go from or basically you reach this particular cell from a variety of directions. So diagonal direction, a vertical jump, a horizontal jump from above and left side also. And you do these calculations and then you figure out which of these scores is the better one. Okay. So the best score, the most positive score among those, that is what you put inside of this cell. So if it is the match or mismatch score, you put that. If it is the gap score, you put that. So from counting from all of these directions, you put the best scores basically. And now you can basically calculate all of the values for all of the available cells inside of the square. And the alignment score for Needleman-Wunsch, that will be the bottom left corner. Okay, so the bottom left corner here it says 2. So in this case, the net alignment score that would have been 2. 2. And one other thing that you need to do here is that whenever you place a value in a cell, you have to trace from which direction it came, basically, from which cell it was derived. It was a diagonal or upwards kind of vertical jump or horizontal jump. You have to indicate that using arrows. And ultimately, if you trace the arrows from the net alignment score, which is 2 in this case, so you can figure out the path of the kind of the alignment, right? So for example, in this case, if you see the 2 value is your net alignment score. So if I just go for the pen, so you will see that if you trace that back, you get this sort of direction from the initialization, which was 0, right? It seems that there are two different paths, which means that you can have two different alignments. And how you can align is basically the diagonal parts, they are of course matching, right? They are matches. For example, this diagonal you see ATG on both hand side, right? So not ATG, so ATG was on the upper hand side. On the left hand side, again the TG was matching because you have to include the gap also here. But how you do it basically is that just beginning from a particular side, you just start to align them. So for example, the first two CA, they are forming a diagonal here. So of course they are matching, right? But the next case, you see a jump here. Okay. So the jump from 2 to 0, this vertical jump. So that means that one character of the left hand side sequence that increased, but at that same time, no character had undergone a step forward on the upper hand side sequence. So in this upper hand side sequence, you will see that there will be a gap present here basically. On the other sequence, the left hand side sequence, so you will see the sequence jump to one character forward basically. And then you continue from that again. So the TG, they are matching. So this T and G part, they are the diagonal part, they are matching. And then it again diverge into two different paths. One is a continuing diagonal where the T versus T is also matched, which is in the first sequence alignment. And then there is a gap present, and another is straight up a gap present, and then a diagonal. Okay. So by tracing all of these paths, you get two different answers to what the sequence alignment could be. And the alignment score is, of course, positive 2 in this case. So based on that, again, you can perform different calculations. Of course, you can encounter them in your exams also. So what you have to do is basically you have to initialize the algorithm with 0. Just continue adding up the gap penalties, continue accumulating the gap penalties on the vertical and horizontal side. And based on that, you can calculate the score of the first cell from the 3 different directions. And if you have the score of this cell also, then you can continue forward by calculating the different cells. And then ultimately, at some point, you will reach X, right? So it will give you the value of this particular cell. Okay. And then we also saw something called the Smith-Waterman alignment algorithm, right? So Smith-Waterman is a local alignment algorithm. Now, in the case of the local alignment algorithm, it is the rule that instead of putting any negative value whenever it is arising, you just put a value of 0 there, right? You put a value of 0, and the calculation process is kind of the same. So you calculate the value of for a particular cell from a diagonal, as well as a jump from the upper cell, as well as a jump from the left hand side cell, so horizontal and vertical, which are gaps, of course, and the diagonal is match or mismatch. You calculate these values, you put the values, but whenever you encounter something negative, you just put a 0 instead of that negative value. And this will help you identify basically a small subsequence of the among this kind of the whole stretch of sequence, which will be aligning together. Okay. So I think we'll just discuss from the local alignment algorithms in the next kind of class. So the local alignment and the rest of the sequence alignment concepts like phylogeny and construction of phylogenetic trees. So we'll try to discuss wrap up in the next class. And then we'll go for the structural part of the protein also. We'll try to recap that as well. Okay. Yeah. So I think in this particular session, I'll just wrap this up. So again, as I always say, for the exams, just consider Professor Gomiha, his lecture series and what he goes on and what it teaches basically in all of these lectures, and the related concepts and the assignment concepts and the similar types of questions that you have also discussed in the TL classes. So those are definitely the type of questions that you will encounter for your exam also. Okay. So yeah. So for today, I will wrap up the class. So thank you everyone for joining. The next class is scheduled just in this Monday. So which will be an extra class or an additional class from NPTEL. So we'll wrap up the rest of it then. Okay. Thank you everyone for joining, and all the best for the preparation for the exam. Okay. Thank you.
Thank you, sir.